Welcome back to the lecture series in cell culture. So, in the last class, we summarized the three techniques of cell separation, where we talked about density gradient centrifugation, we talked about immunopanning, and we talked about fluorescent assisted cell sorting. Now, coming back, one small tail piece which I didn't cover while we talked about the immunopanning of the motor neurons, which I just wanted to tell you how powerful these techniques are, provided you understand the development biology of most of these different cell types. So, in the embryonic motor neurons which are present in the ventral horn, if you remember that I showed you that diagram, I am just going to go back. Yes, in the ventral horn motor neurons, where I told you that you have two types, like you have the larger size, you have the smaller size, but what is important here I wanted to highlight is that these ventral horn motor neurons, these large motor neurons express that unique extracellular or unique antibody at only or at specifically at around E14, embryonic day 14, and after that all the cells here express it. It's something like that, I will show you. So, we are into week 7 lecture 2. So, say for example, this is the spinal cord, okay, and this is the ventral horn. The shaded region is the ventral horn. Ventral horn, and this is the dorsal horn. Dorsal horn. Now, interestingly, so the embryonic day starts from say E0 to say E22, when this is actually not called embryonic, it is called fetal. Okay. So, embryonic day lasts till, so this is halfway through, okay. So around 11, this is also called embryonic, and up to 14 it is called embryonic day, 14, 15, after post 15 it is called fetus. Okay. So, around I would say E15 and then I would say F18 likewise. Maybe you can consider from 18th day it is called a fetus. It is why it is called a fetus is at that particular time the whole anatomical features comes out perfect. Prior to that, the whole anatomical development was kind of not, you cannot really fully make out. It is there, you know that this is a small pup or something, but not fully. Okay, that is why these two distinctions are there. So, coming back, when we talk to you about separation of embryonic motor neurons, so this is what we are kind of revisiting this problem in the light of our new techniques, what we have learned, cell sorting by immunopanning, density gradient and fluorescent assisted cell sorting, embryonic motor neuron isolation. Okay. Now, so these motor neurons are sitting here, right. So, now within this there are many other cells which are sitting there. Okay. Now, what is unique about the motor neuron at E14 or mostly E14 you can think about is they express a very, very unique marker which cannot be seen in any one of them. Like this green is that marker what I am. This is at E14, the story at E14. Only these cells are expressing this marker. But as time progresses, as we move from F18 and likewise 16, 17, then this antibody or this marker, not antibody, this marker is expressed all over. Why I wanted to highlight this point? This deserves a different highlight because of the reason that unless you exactly know the developmental paradigm of a particular tissue type, mark my word, you should not jump onto cell sorting. First of all, you try to understand what you are doing. If the specific antibody is there and you have or a specific marker is there, sorry, pardon my language, it is not antibody, it is the specific marker is there on the cell surface and you have a counter antibody to bind to it, then only you should go for it. But say for example, if somebody uses this paradigm which is supposed to be followed at E15, you try to use it at E18, it's just not going to work. I mean, you will get a lot of contaminating other cell types, right. So, these are some of the points what I wish that you people should invest your energy whenever you are doing cell sorting. You should read the literature thoroughly, that how things have developed, what others have done and where you are, where you want to take it to the next level. What are those critical questions what you are trying to address. So, it is very important that you go through this whole roller coaster ride of understanding that why it is so significant, that you understand. That is why I kind of revisited this, that these profound tools can come very handy provided you know the power of these tools. If you do not understand the power of this tool, you can misuse a tool, which many a times I see and which saddens me, but that is how it is. Okay. So, having said this, now I will switch gears from cell sorting part to a next level. Okay. That is, in a culture dish, when you grow cells, what is our anticipation? Our anticipation is that it should not only maintain its phenotype, the cell should not only maintain its phenotype, it should be able to maintain its functionality as far as it can afford. So, having said this, say for example, we wanted to culture a cell which secretes insulin. Now, we have to ensure that those cells in the culture dish indeed secrete insulin, may not be with the same intensity with which it does in the body, but maybe 50% of that or maybe 10% of that, maybe even 5% of that, I will accept it, or maybe even 2% of that, but it has to continue with that function. Or say for example, I say I wanted to culture neurons, great. Now, what is the function of a neuron? It is a electrically active cell. So, electrically active cells means it fires action potentials. Now, if I have neurons which does not fire action potential in the culture dish, or if I claim that from XYZ stem cells I have derived neurons in the culture with all possible immune markers I show it. For me, it won't make any sense unless otherwise it has been shown that yes, indeed, those neurons what I have cultured are electrically active. If they are not, then boy, there is a problem. There is some serious issues out here. And it is very important that whenever we venture into the world of cell culture, one has to keep this basic fundamental ideas in mind, that what are you targeting, what is the goal. So, say for example, whenever I ask this thing, so the critical question, let us put the critical question first. Okay. So, the critical question is that has the regenerated, because you are regenerating the cells in the culture, regenerated cell type with, if I say with special reference to neurons, I take the liberty with a special reference to neurons retained the original phenotype or close enough phenotype as experienced by the cell in its native environment. Okay. So, when we talk about native environment, so we are talking about something like this. So, let us repeat it. Has the regenerated cell type with a special, of course, we are talking about the neurons in this situation, which could be any other cell type with a special reference to neurons or other cells, retained the original phenotype, this is part 1, or close enough phenotype experienced by the cell in its native environment. So, this is part 1, the phenotype. So, what we mean by phenotype here is, say for example, I say this cell is destined to become neuron. So, I will expect that this cell will have a processes which will have an axon, dendritic tree, and it will express all the necessary ion channels to behave like a neuron. So, in other words, it should be able to shoot an action potential. Similarly, if I say this cell is, this community of cells what I am growing is supposed to secrete insulin, then I expect that they will form this kind of clustering, and I should be able to see they are secreting insulin, which of course I have to quantify by some kind of titration or whatever, right. So, here apart from the phenotype, I introduce another aspect which is called functional. So, morphology plus function. Could these two parameters being retained in the cell culture dish? Unless this is being retained, the purpose will not be achieved, of having a model which is close enough. So, having said this, and again as I told you that I will be taking mostly the case of neurons here, because they are the most challenging ones. So, for example, again if I take another excitable cell, I say I am culturing cardiomyocytes. You know what will be my first question? Are they beating? Are those cells in the dish beating? Are they contracting like that? If they are not, then I will have doubt that are you really regaining the phenotype as well as the functionality. They look like great like phenotypes, yeah, sure, but then are they functionally active? If they are not, it means we have to do something. Similarly, if you culture a muscle, say skeletal muscle, my first question again will be, could you stimulate it so that it contracts? Because all our skeletal muscle, what we have, they have both electrical as well as mechanical function. Are we there? Or I will ask a question that, okay, you are growing the muscle. Say for example, let us take the example of muscles. So, generally whenever we talk about muscle culture, we talked about there are two possible ways, and here we will talk about say skeletal muscle. Let us take the example of skeletal muscle first. Okay. So, mostly skeletal muscle could be cultured from two different sources. One source is your E18, sorry, F18 rat fetus or mouse fetus. E8, it could be F18, it could be F20, or it could be a neonatal, which is day 1 to day 3. Should not exceed, because it is really challenging to isolate the tissues and dissociate it. Or you do it from satellite cells obtained from adult hind limb. And the reason why they follow hind limb, so here also this is all from the hind limb. Hind limb. The reason they do it from the hind limb is that you get sufficient amount of tissue. Say for example, so it's something like this. If okay, so basically the tissue is collected, and if it is a fetus, it will be something like this, and if it's an adult, okay. So this is the head. Similarly, here is the head, and here you have the hind limb. So most of the tissue is collected from here. Now, the satellite cell, if we talk about this as an adult, so satellite cells are sitting like this. If this is the tissue which is making the hind limb, underneath out here, and these satellite cells are collected. So these satellite cells are essentially stem cells destined to become a skeletal muscle. And suppose you have to isolate these satellite cells from here, you needed to do a lot of the techniques what you have read, cell digestion or enzymatic digestion, enzymatic / mechanical digestion 1. Second, what you have to do is, followed by that, after this mechanical and enzymatic digestion, next step will be cell sorting. Once you sort out these cells, then you have to you may have to use FACS or some other density gradient or FACS or some kind of immune marker to isolate these satellite cells, and then you have to grow them. So, this is a long route where you have to grow from the adult satellite cells, and again when you grow it from the adult satellite cells, your success rate is not that easy. It is kind of tricky. Now, you might wonder why we needed to use the satellite cells. There are reasons for it, and that is where I am going to come, that why you cannot use the other cell type, and that is precisely is where we will be discussing about skeletal muscle, that how they grow and how you have to be careful while we will be doing this kind of cultures. Okay. So, I will close in here. In the next class we will follow it up from here, that how to isolate these cells and what are the phenotypes we see and what are the functional analogues of it. Thank you.